Hello everyone, and welcome to this overview of assaying ternary complex formation for targeted protein degradation with your FIDA. In this video, we'll be going through the experimental setup as well as the subsequent data analysis. But first, a brief reminder on the terminology and FIDA assay setups, as we will be using multiple different strategies for this type of assay. A FIDA experiment measures the hydrodynamic radius of a fluorescent molecule by characterizing its diffusivity. Two important FIDA terms here are indicator and analyte. The indicator is the fluorescent molecules whose size is measured, and the analyte is the compound that is titrated. There are four ways of doing a FIDA experiment: premix, capillary mix, capillary dissociation, and capillary flow experiments. With premix, you have an equal amount of indicator and analyte vials, and all of the samples are premixed prior to injection. Using capillary mix or cap mix, you have a single indicator vial and a titration of analyte vials. Using this method, the samples are mixed in the capillary by the FIDA instrument. When doing capillary dissociation or cap dis, you have a titration of mixed indicator and analyte vials and a single buffer vial. The complex is then injected into buffer. Capillary flow experiments or CapFlex you can ignore unless doing liquid-liquid phase separation and condensate analysis. In targeted protein degradation, the goal is to use a small molecule to facilitate the formation of a ternary complex between a target protein and the ubiquitin E3 ligase. This causes the target protein to be ubiquitinated and subsequently degraded by the proteasomal pathway. One way of achieving this is to use bispecific molecules. Since the ternary complex formation is governed by two equilibrium constants and their cooperativity, there is a Goldilocks zone where the maximum amount of ternary complex is formed. All biophysical parameters relevant to this complex formation can be characterized by FIDA. Another approach is to use molecular glues. These modify the surface of the target protein to facilitate the interaction with the E3 ligase. As there is only one affinity constant for the small molecule, there is no hook effect, and the assay is performed like a standard FIDA KD determination. The experimental setup for a molecular glue is very similar to a standard FIDA assay. It can be performed as a cap mix or a premix assay. These kinds of assays can be performed directly in cell lysate. If you want to perform your assay directly in cell lysate, a premix experiment is recommended. There you will have an equal amount of analyte and indicator vials. The indicator is of course mixed with analyte. An example of ternary complex formation directly in cell lysates can be seen on the left. The three indicators on the left and the size of the ternary complex can be seen on the right. No change to your standard FIDA methods are required, and the experiment can of course be performed as complete walkaway automation. To do full characterization of ternary complex formation using bispecific molecules, you need to make a titration of bispecific molecule with a constant concentration of labeled target and excess of unlabeled E3 ligase. This kind of experiment can be performed as a cap mix or a premix experiment. The cap mix experiment, you have one indicator vial and a titration of analyte vials, while in the premix experiment, you have equal amount of indicator vials and analyte vials. All relevant biophysical parameters for your systems can be characterized by this method. This includes the cooperativity. An example of this kind of experiment is shown on the left side. Please note that it is possible to overshoot the ternary complex formation with too high concentration of bispecific molecule. This is due to the hook effect. As such, it is recommended to do a few initial titration points to verify the correct concentration range prior to setting up the full cut titration. No change to your standard FIDA methods should be required. Once you have run your experiments on the FIDA instrument, you can analyze the data using the FIDA software. Going to the Data Analysis tab of the software, you can click the Load Data Files button and a Pathfinder window will pop up where you can navigate to your data folder and select the data you want to analyze. Once you have loaded your data, you can then evaluate the quality of your experiment. You select the time interval that will be fitted by selecting Data Start and Data End. If the baseline is drifting, this can be fixed by toggling the relevant tick. The noise reduction can also be toggled on and off. You fit your data by clicking single species or multi-species. In this case, there are 2 species, so we will use this fit. You can save the result by clicking the relevant button. This procedure is then applied to all data points. If you don't want to manually fit every last data point you have recorded, you can select the Auto mode. If you select the Auto mode, the top of the analysis software changes to an overview of the parameters that can be applied to your fit. You select the relevant fitting parameters, in this case a single species fit with standard settings. Click Load Data Files and go. This causes a pop-up where you can select the relevant files. Then the software calculates for a bit, and when it is done, it shows you the analyzed data in the Binding Curve tab. By clicking the Data Analysis tab, every fit the software has done can be manually evaluated and corrected. In this case, a correction is required. You see here that there has been an air bubble in the middle of the tailogram. You can also see that the R square is marked in red. This data point can simply be deleted, and you can click Generate Binding Curve to see the new binding curve. This updates the Binding Curve tab. Now you have your data, you can viscosity compensate by clicking this tab. To fit the data, you have to select the ternary complex model in the Select Model tab. When you do, you will see a pop-up asking for a few initial guesses for the parameters. The size of the indicator should be set to the data point at a concentration of bispecific molecule of 0 micromolar. The binary complex size is the end of the titration curve. The size of the ternary complex is the top of the titration curve. KD1 and KD2 should be set to the half point on either side of the top point of the curve. A good initial guess for the cooperativity is neutral, i.e. a value of 1. Lastly, you need to tell the software the concentration of unlabeled protein in your assay. You then click Apply and click Fit Binding Curve. The results are then displayed on the right-hand side of the screen. We see that this bispecific molecule has a very high affinity for both its target proteins and has a positive cooperativity. In this model, a cooperativity bigger than 1 is positive, and a cooperativity smaller than 1 is negative. That concludes this video on ternary complex characterization with targeted protein degradation. I hope you found it helpful and enjoy your assays.